Good afternoon and welcome to the MGI webinar on non-model organism sequencing using MGI DNBC platform. I'm Jiyoti Verma, Field Application Scientist at Decode Science, and I will be hosting this webinar. So before we start, I would like to mention a few things about Decode Science. We are distributor of MGI products in Australia and New Zealand. Next slide, please. Besides MGI, we also supply all these high-quality leading-edge technology brands and provide technical and application support for all these brands. Next slide. And we have an excellent sales team. Our business development managers are based in different cities, so even during lockdowns, we are able to provide exceptional support to our customers. You can find more details about us at our website. Next slide. So this is the outline of today's webinar. I'll start with a short intro of DNBSEQ platform, and then we have two main talks. First one is on metatranscriptomics of sponge microbiota by Dr. Paul O'Brien from James Cook University, followed by 5-minute Q&A session. And the second talk is on Macadamia genome de novo assembly by Professor Robert Henry from University of Queensland, followed by Q&A session. So for Q&A session, you can type your questions in a Q&A box, which is present at the bottom of the screen. And we are running a poll today. So poll box also you can find somewhere at the bottom of this screen. And we would appreciate if you could participate in this poll during this webinar. And we also have a quick survey at the end of this webinar. So let's start with intro. Can you change the slide? Yeah. So today's introduction, I'll explain about key principle of MGI DNBSEQ technology and how MGI DNBSEQ platform may serve as an alternative to the market-leading platform from Illumina. Next slide. So this slide shows the technical comparison of MGI DNBSEQ and Illumina platform. In both platforms, first DNA molecules are fragmented and adapters are ligated to the fragments and processed to produce libraries. So DNBSEQ library has single-stranded DNA circles with adapter in the middle, and Illumina library has a linear double-stranded DNA with adapters at both ends. So these libraries are then replicated to produce an amplified signal for sequencing using rolling circle amplification, producing DNA nanoballs for DNBSEQ, and bridge PCR amplification, producing clusters for Illumina. Both platforms then use a stepwise sequencing by polymerase on a single-stranded DNA template with fluorescently labeled nucleotides. So the exponential amplification performed on Illumina libraries during cluster generation step is known to have issue with molecular barcode switching and GC-rich sequence coverage. So in next few slides, I'll explain how MGI DNBSEQ platform has been successful in reducing some of these issues. Next slide. So this slide shows the circularization step following PCR amplification. During this step, double-stranded DNA is denatured by heating to single-stranded DNA, and then you add a splint oligo, which helps in circularization of the single-stranded DNA, which has complementary sequence to the splint oligo present at both ends. So as a result, you get single-stranded circular DNA, which is our final MGI library. Next. To amplify the signal for sequencing, then we make DNA nanoball from this single-stranded circular DNA library by a rolling circle amplification reaction. So to perform RCA reaction, you add primers, which binds to the adapter region, and use a high fidelity DNA polymerase to generate up to 300 to 500 copies of your original template. And because only the original DNA template is used to generate copies here, amplification errors do not accumulate and resulting in high accuracy for detection. And the small size of DNBSEQ results in smaller and more concentrated DNA spot fluorescence, giving DNBSEQ a higher to signal noise ratio and faster sequencing times. Next slide. Yeah. DNBSEQ uses pattern array technology that has flow cell with uniformly placed active spot to bind DNB, and each active spot is only large enough to bind a single DNB. So when you load DNB into the flow cell using loading buffer, DNB carry a negative charge and active spot on flow cell carry positive charge. So this negative and positive charge is the main driving force behind DNB binding onto the flow cell chip surface. And the proprietary loading buffer can further ensure DNBs sticking onto the same spot for hundreds of cycles without any compromise signal. So this results in high sequencing accuracy with no index hopping, high chip utilization, and an average duplication rate below 3%. Next slide, please. So for sequencing, DNBSEQ platform utilizes the CPARS technology as shown here. The CPARS consists of 3 steps. First step is incorporation of a fluorescently labeled DNTPs with a DNA polymerase and washing of any unbound DNTPs. So in step 2, image of DNB flow cell is captured and fluorescent signal is converted to digital signal, and the base information is determined using MGI proprietary base calling software. So after the image is taken, cleavage of fluorescent dye is done in step 3, and this prepares the DNB for next cycle. So one CPARS cycle can detect 1 base. So if you want to read 100 base pair of your library, this cycle must be repeated 100 times. Next slide. So this is the take-home message from these what I discussed so far. And DNBSEQ technology has 3 main features: DNA nanowall, pattern array flow cell, and CPARS technology. So when you combine all this together, you get increased accuracy, decreased duplicates, and low index hopping in your sequencing data. Next. So I'll explain briefly what is Sdlfr. As the name suggests, it's a single tube reaction. It is based on DNA barcoding method invented by Chief Scientific Officers of MGI in 2012. And here you add same barcode to all subfragments of the original long DNA. So this gives you long-range sequence information from short reads, which is very cost-effective approach to genome assembly. So next slide shows the Sdlfr. This slide shows the Sdlfr workflow. Here you start with 1 nanogram high molecular weight genomic DNA and do transposon insertion along the length of this long DNA fragment. And then you add cloner barcoded bead library, which has around 3.6 billion unique barcodes. And each bead surface has around 400,000 copies of the same barcode. So then you hybridize DNA to these beads. And since we use higher beads to DNA ratio, this ensures we only have 1 DNA molecule per bead. So then we do ligation of barcode oligo to insertion to transposon inserted sequence using a splint oligo on the surface of the bead. And next, we remove transposase, digest extra oligo, and then add second adapter followed by PCR amplification and circularization to get single-stranded circular DNA library and make DNA nanowall and finally sequence it. Next. So with traditional whole genome sequencing without the molecular barcoding, mapping can be difficult, especially with shotgun sequencing. You can try to physically separate your fragments, but it's not a precise method. However, in Sdlfr, that is based on DNA barcoding method, you add same barcode to all subfragment of the original long DNA. So this can help speed up mapping and genome assembly. So besides genome assembly, Sdlfr works well for techniques like haplotype phasing and detection of structural variation such as indel, translocation, and inversion. And you will hear more about Sdlfr in Professor Robert Henry's talk. Next slide. So this slide shows range of MGI products that can support entire NGS workflow from sample collection to final report. And this include easy to use automated sample transfer and sample preparation system and high throughput sequencer, various extraction, library prep and sequencing kit, and data analysis software. So next. So if you want to get access to MGI sequencing platform in Queensland, there are two places: Central Analytical Research Facility, also known as CAF, at QUT. This facility has high throughput G400 sequencer, which can support a range of read lengths. And if you are interested in much larger scale sequencing project, you can get access to the T7 sequencer at MGI Australia Demo Lab in Brisbane, which has ultra high throughput and high speed sequencing capability. Next slide. So right now we are taking application for MGI DNBSEQ and PS research grant valued up to $30,000 each, and submission deadline is on 8th October. So if you are interested, you can find further details at our website. So that's all I have for intro. Thank you so much. And let's move to the first talk of today's webinar. So I would like to invite Dr. Paul O'Brien from James Cook University. Today he will present Characterizing the Sponge Microbiota using Genome-Centred Metatranscriptomics.
Thank you. And yeah, hello everyone. Thanks for inviting me to speak today. So yeah, as was just mentioned, I'm going to talk a bit about some of the work I did during my PhD, which was in collaboration with BGI. So the whole project is kind of based around host microbe symbiosis and coevolution in coral reef invertebrates. But I'll just talk specifically about some of the genomic work we did with sponges. I have now moved on to a new role, so now I'm working at the University of Queensland under the Australian Centre for Ecogenomics. But for today, I'll talk about some of the stuff I was doing during my PhD. So when we think about coral reefs, I guess intuitively you think about the corals and the reef fish that inhabit those reefs, some of the more colourful and vibrant characters there, but I guess a lot less focus is on the sponges. And these are actually really important reef organisms. So not only are they really abundant and really diverse, I think there's around 8,000 described species, but they're really key members of coral reef ecosystems. And that's partly because of this ecological function that they have. So they're filtering these really large volumes of seawater. And when they do that, they actually are assimilating dissolved organic matter. So they're providing this really important trophic link. So coral reefs are typically nutrient poor environments. So when sponges are filtering that seawater, they're allowing some of that dissolved organic matter to be used by other reef organisms. So for example, seaweed and coral, for example, can release dissolved organic matter such as carbon into the seawater, which is generally unavailable for most of the reef organisms, except for maybe the microbes. So sponges can actually take up that dissolved organic matter and assimilate that. And then when they shed their cells as detritus, this can then be fed on by the detritivores and make its way up the food chain. So they are providing this really important trophic link. And so they have this important ecological role on coral reefs. But part of that is actually fulfilled by the microbes that inhabit the sponge. So in particular, some microbes have really dense some sponges have really dense microbial communities. And this role can actually be fulfilled by somewhere up to 80-something% of that dissolved organic matter can be assimilated by the microorganisms inhabiting the sponge. In some cases, it's a lot less depending on the abundance of the microbes there. So we really would have an interest to try and understand how these microbes are interacting with their host. We are starting to get a fairly good idea. A lot of that work has been done through metagenomics. So if you're unfamiliar with metagenomics, basically we're talking about those genome sequences of a community of organisms that inhabit a particular environment. And this can be really useful for studying some complex microbial communities. For example, that's of a sponge. So I know there's a lot in this figure. Basically, I just want to point out that this is one sponge that is being quite well characterized using metagenomics. For example, there's potential there for different carbon fixation pathways. There's a lot of potential there for different microorganisms to biosynthesize B vitamins, so they could potentially supplement that from the sponge diet. There's a lot of microbes there that are involved in different types of sulfur metabolism. and as well as nitrogen metabolism. So looking at nitrification, denitrification, and nitrate reduction. So I say potential, that's because when we're looking at these genome sequences, we can only really infer from those coding regions what those macros might be doing. So what we wanted to do was kind of take that a step further and look at metatranscriptomics. So that will then show us which of those genes are being expressed. So the aim of that kind of section, or that chapter of my PhD, and what I'll focus on today, was trying to get an overview of microbial metabolism in sponges using this method of genome-centred metatranscriptomics. So I said I'll be focusing on sponges. I just wanted to point out that we have done some of the work and the sequencing for corals and octocorals as well. But the analysis is just a lot further along for the sponges, so I'll focus on that for today. So we collected 3 different species of sponges. So these are pretty common sponges that you'll see on the Great Barrier Reef. This is Yersinia ramosa, Yersinia microcondylosa, and Phylospongia, and we sequenced the metagenomes of those 3 species. We then did parallel sequencing for the metatranscriptomes of a subset of those species. So this was Yersinia microcondylosa and Phylospongia foliescens. So just a quick overview of the lab methods that we used. So for the metagenomics, to take our sponge sample, we then want to enrich the microbial cells. So when we sequence, we're not sequencing all the host as well. So basically we're doing a bit of cell separation. This is through centrifugation and some filtering steps to get our enriched microbial sample. Then it's just DNA extraction, and we sequence that with our collaborators at BGI. For the metatranscriptomics, so we take the same sponge sample and then extracted the RNA. We then needed to enrich for microbial mRNA. So first thing we do is try and remove as much ribosomal RNA as possible, and then try and remove as much eukaryote mRNA as possible. By that stage, you only have a little bit left of mRNA, which is hopefully mostly microbial mRNA. We then amplify that back up, and then we synthesize that to cDNA, and again, we sequence that with our partners over at BGI. So just a couple of quick notes on the sequencing process. So we used obviously the DNBC platform on the MGI C2000, although I believe that's now changed just the name, and we used just 100 base pair paired-end sequencing. So we actually got quite a lot of data for our metagenomes, probably is quite a lot compared to what is normally done for this sort of study. So for each metagenome, for each sample, we sequenced 25 to 30 gigabase pairs of data for each sample. Then for the parallel metatranscriptomic sequencing, we went a little deeper, and that was because there's still quite a lot of ribosomal contamination there, so it's really hard to remove that in the laboratory. So we sequenced about 50 to 60 gigabase pairs of data for the metatranscriptomes. Just a quick overview of the Bio Informatics. If you want to know more, happy to answer any questions on that. So we assembled each sample separately. So this was using Metaspade, so de novo metagenomic assembler. Once we had our assembled contigs, we then bin each genome into kind of putative genomes. So these are called metagenome assembled genomes, or MAGs. And did this using about 6 different methods, using 6 different binning methods, and then basically took the best bins of each method to get a final non-redundant set of bins. And then from there, can do our genome classification, looking at different genome statistics as well. For the metatranscriptomics, first removed any remaining ribosomal RNA. We then used our genomes from our metagenomic analysis as kind of our reference genomes, and then we mapped our mRNA reads to those genomes. And then from there, we can annotate our genomes and quantify the number of reads mapping to each gene in our genomes. So first, just want to show you some of the results from our metagenomic binning. So this was across the 3 species of sponges, and we got 1,290 metagenome assembled genomes, or MAGs. And so our cutoff here was for 50% complete and less than 10% contamination, which is kind of like the minimum that you would use. Probably most of those will, I'd say, probably greater than 80% complete. So since we assemble each sample separately, we then have to de-replicate them. So it's likely that you'll assemble the same genome in different samples. So we first compiled all those genomes together and then de-replicated them at 95 average nucleotide identity. And from there we had 415 kind of unique genomes. So this is just a phylogenetic tree of those genomes that we assembled. So in the middle is the tree, and then in the inner ring we have each clade is colored by the taxonomy of the microbes. So that's the phylum level. And then the outer ring is showing you a heat map of the relative abundance of those genomes within each sponge. So altogether, we assembled 21 bacteriophyla and 1 archaea phylum as well. So just a couple of quick things on that tree. So we had that 1 archaea phylum, which is the Microbiota, but this was only present in Yersinia ramosa, and the other, it was absent from, or at least we didn't find it in the other 2 sponges. We also saw quite a high abundance of cyanobacteria. In particular, there was one genome in particular in Yersinia ramosa, which is the outer samples, and one genome in particular for the inner samples, which is Phylospongia foliescens. And we also saw quite a high diversity of proteobacteria. So around a third of those genomes were classified as proteobacteria. So this is all pretty common, or what you would expect to see from these sorts of sponges. So we had good representation of the microbiome there. And just a quick overview of the metatranscriptome mapping. So for Phylospongia, we had around 150,000 total expressed genes. And most of these were actually from cyanobacteria. So it was quite an abundant phylum, is one genome in particular. And around 70 to 75% of gene expression was coming from cyanobacteria. So this was the genus Synechococcus. And to a lesser extent, we saw quite a bit of expression from proteobacteria as well. For our Yersinia samples, we had around 400,000 expressed genes, but this was far more diverse among the different genomes in that sponge. So again, quite a lot of proteobacteria, and to a lesser extent, porobacteria, which is a kind of specific sponge clade. But overall, we had quite a large spread of gene expression across the different members of the Yersinia microbiome. So the next thing we did was look into a range of different metabolic pathways. I won't show you all of them because, try and keep it within time. But basically, we looked at carbon fixation, sulfur metabolism, nitrogen metabolism, and a few other things as well, vitamin synthesis, amino acid synthesis as well. So for carbon fixation, we checked about 6 different autotrophic carbon fixation pathways. So this heat map here is showing on the y-axis, we have the taxonomy of the different genomes, and this is grouped by microbial family, just to simplify the figure a bit. And then on the x-axis, we have the different genes that are associated with the different pathways. And then that's faceted by each step within each reaction pathway. So for carbon fixation, I guess unsurprisingly, we found that this was the carbon cycle was fully expressed by the cyanobacteria Synechococcus. So across all the different pathways that we looked at, this was the one that was fully expressed. So it kind of suggests that these cyanobacteria could supplement some of the carbon needs for the sponge. And we saw the same thing with Yersinia microcondylosa as well. So this function of cyanobacteria seems to be common amongst these 2 sponges, and has been shown in some of the metagenomes of other species as well. And the next thing we looked at was sulfur metabolism. So one of the interesting things about sulfur is, I guess in sponges and other animals as well, there's the host produced taurine. So taurine is a form of sulfur. And we found that both different alpha proteobacteria genomes and gamma proteobacteria genomes were expressing the necessary genes to kind of import that sulfur into the cell and then oxidize it to sulfate. So this was amongst the alpha proteobacteria and gamma proteobacteria mags. We also looked at sulfate reduction to sulfite and sulfide, and found that gamma proteobacteria mags were again expressing the necessary genes for sulfate reduction and thiosulfate oxidation as well. So this produces sulfate, and we found that alpha proteobacteria was expressing the full set of genes for thiosulfate oxidation. And to a lesser extent, gamma proteobacteria, although as you can see, it was missing one of the key genes there. This was similar for Yersinia again. So we had taurine metabolism, again with alpha proteobacteria and gamma proteobacteria genomes. And we also saw nitrospinota genomes showing taurine metabolism as well. And then for the sulfate reduction and thiosulfate oxidation, we again saw that with the gamma proteobacteria. And the last thing I just want to show you is some results on the nitrogen metabolism. So we looked at nitrification, denitrification, and nitrate reduction, and actually most of the nitrogen metabolism in the microbiome of Phylospongia was absent. So we didn't get the full pathway for any of those nitrogen metabolism processes. What we did see though was some expression of nitrate reduction, so to produce nitrite. And we also saw expression of nitrous oxide reduction, which is not commonly seen in sponges, so that was interesting. For Yersinia microcondylosa, those three processes, nitrification, denitrification, and nitrate reduction were mostly complete in this sponge. So we did see a lot more nitrogen metabolism happening here. But although it was quite varied amongst the different microbial lineages. So I'll just point out a couple of things. So one thing we found was that ammonia oxidation, which is generally a limiting step in nitrification, at least in sponge microbes, almost had the full expression in Sdlfr bacteria. So we had the 3 genes there, and 2 of them we found expressed. And this is normally confined to the Macrobiota in sponges. This sponge, at least we didn't find Macrobiota in this sponge, so it could be that Sdlfr bacteria actually performing ammonia oxidation there. We also, similar to the other sponge, saw nitrate reduction and some other processes of denitrification and nitrate reduction. And again, we also saw the nitrous oxide reduction as well, so to complete that process of denitrification. So just want to give just a quick overview of kind of a summary of those results. So we had, so it was our cyanobacteria across both the sponges that were showing carbon fixation. So it's of high potential for that to then be used by the sponge. We saw that proteobacteria across both of the sponges were highly involved in different parts of sulfur metabolism. And nitrogen metabolism was mainly confined to our Yersinia microcondylosa sponge. And it was the Sdlfr bacteria there that was providing some of those unique functions. And so that's it for today. So my supervisors during my PhD were David Bourne, Nicole Webster, and David Miller. And our collaborators over at BGI were Gaoji Zhang and Shangjing Tan, who helped me quite a bit with the bioinformatics analysis. So thank you very much. Well, I think you're muted. Can't hear you.
Thank you, Paul. And we have one question for you in the Q&A box. So what kind of library prep kit did you use? And is it from MGI or third party?
Yeah, so my role was to get all the DNA and RNA ready for sequencing, and then I sent that to our BGI collaborators who did the library preparation. It was the MGI kit that they used. I can't remember exactly which one, but yeah, if you want, feel free to send me an email afterwards and I can get back to you on that, but I can't remember off the top of my head, sorry.
Okay. Yeah. That's it. There are no more questions for you. So we can start the next talk. Thank you, Paul.
No worries.
So for the next talk, I would like to invite Professor Robert Henry from University of Queensland and today he will talk about Macadamia genome de novo assembly. So welcome, Robert.
Thank you. I'll just share my screen. So today I'm going to talk about our work with macadamia genome sequencing, and this has been a relatively part of a longer term collaboration with MGI on sequencing a number of different plants. And this is formed part of a much larger project sequencing the genomes of, working on the genomes of horticultural tree crops in Australia. These 5 species represent most of the value of horticultural tree crops in Australia, and the avocado, mango, macadamia and citrus are particularly important in Queensland as tropical and subtropical crops. So we're working on detailed genomics of these species as part of the broader program. With macadamia, we have 4 species, all found here in this part of the world, really centred on Brisbane. We have a couple of species that have formed part of the domesticated macadamia and a couple of others that are purely wild species. All 4 are found still in the wild, but all are rare. One of those species, Macadamia jansenii, which is found only as a very small population in the north, is now an endangered species with only around 200 individuals known. And so we've chosen that as a model species to use in a lot of our genome work. It's a good example of a typical plant with a genome of modest size, and it is relatively homozygous because it's rare. It doesn't have a lot of heterozygosity, which we find in many of the other species. So we've used this as a model really to compare a wide range of different sequencing techniques, both the sequencing and assembly tools that are available, and much of this we've done in collaboration with the Genome Innovation Hub here at UQ. And if you want to look at the primary comparisons, and these go back a little while, this would be 2019 work, and it's important to think about the date because all these technologies keep improving and changing. But we've got a very good comparison there in this paper in Decode Science that will help you to look at the detail of what I'm going to talk about. We've been publishing many updates on this as new iterations of the technology come out. We're repeating the sequencing of this same sample and showing how the technology is evolving on different platforms. My own interest really in sequencing comes from a long-term involvement in development methods for genotyping plants. All of these books are ones that have come from my lab over the years where we've been continuing to really reflect on the advancement of marker technology and genetics in support of agriculture. But very much the whole focus now is on sequencing. My view is that the best way to genotype an individual is to sequence its genome, and the genome sequence is the best genotype we're likely to get for that genome. And because of the collapse in the cost of sequencing, it must become the preferred method for genotyping in almost all situations, and that's particularly going to be the case in the future. And of course, this means, and this is a BGI photo, I think, that a lot more of our work requires that we develop the capability of working with much larger volumes of data than we have in the past when we were using small amounts of marker data to manage our plant and animal systems. We apply sequencing at sort of 3 levels in plants. And first of those is looking at the organelles, and particularly the chloroplast. The chloroplast has been widely used phylogenetically in plants because it's so highly conserved. But by now, being able to get the complete chloroplast genome sequence for any individual very easily using these sequencing techniques, we can study a whole lot of processes. And what we're seeing is things like reticular evolution in plants is much more widespread we probably understood before, with gene flow between species as well as within them, happening at reasonably high rates right across the environment. We're also very interested in working with transcriptomes, and this is really a key way that we're driving gene discovery for important traits, is looking at differential expression transcripts, but it's also allowing us to look at things like allele-specific expression, which is really important in many contexts in understanding the performance of different organisms. The ultimate, of course, is a nuclear genome, and this is where we really apply these long-read techniques. Gene discovery, of course, is based on us comparing the genomes of different individuals. What's becoming much more important is to have genomes where we can look for the presence and absence of genes, because in many cases, those presence-absence differences within a species explain much of the variation. It's not just allelic variation, it's actually gene variation as well, that requires that we have a genome sequence of every individual. Just to go back briefly to talk about the chloroplast, the sequencing of the chloroplasts with short-read sequencing technique is the preferred approach. Long-read method, the very long-read methods really don't have the accuracy we need. Because the chloroplast is so highly conserved, even a single SNP between individuals is likely to be significant. We only get a very small number of differences between individuals, and so each base is important. We need a completely accurate sequence, and we strive to have a base perfect sequence of the chloroplast of every individual. And we'll be doing this on the MGI platforms quite successfully, because when you sequence the DNA from a plant, you get a very deep sequencing of the chloroplast, when you might only be getting quite a shallow sequencing of the nuclear genome, because the chloroplast is present in 1,000 copies for every nuclear genome copy. So that means that the coverage of the chloroplast is incredibly high, allowing us with our de novo assembly and map-based assembly techniques, we've got a pipeline that allows us to get to a verified, highly accurate chloroplast sequence for each individual from just short-read sequencing. And that's really allowed us to go a long way with understanding plant relationships. And of course, we now have improved annotation of that with software, so we can annotate all of the genes. We know the function now of all of the genes in the chloroplast. It's a completely defined system, a very good one to work with, and to compare any two DNA samples. So any two DNA samples for which you've got an MGI sequence, you can get it of a plant, you can get the chloroplast, and that can be a really great way of identifying it. For long-read sequencing methods, with the Macadamia system, we've really compared all of the available platforms to allow us to understand the pros and cons and strengths and weaknesses of the different approaches. And they all have various strengths and weaknesses. So we've used the Oxford Nanopore and the PacBio versions. And as has been introduced earlier, the BGI single tube long fragment read technology, which I'll focus on talking about today. Complementing that still, of course, very importantly, are the short read methods from Illumina and MGI, and we find both give us really good data, and methods that we use to assemble the chromosomes at a higher level, the high-C methods of chromatin mapping that allow us to put contigs together to make full chromosomes. We're using a version called OmniC that is less biased in the way that it allows us to put those maps together. So I'll just go through some data applying this long-read method to our Macadamia system. And the way I think about this is it's really a pseudo long-read method, because it's using and taking advantage of the accuracy and so forth of short reads. But because we've barcoded those short reads and know which ones come together, we can actually put together reliably a much longer read using that fundamental platform. And so here in this system, we're able to get a 96-fold coverage of the genome. And you know, we've got a price there. It's very important. We compared the prices of all the techniques. This was, we thought, very cost effective for that date in time. The prices will keep changing, of course, but for a long read sequencing at this depth of an organism like this was an incredibly cheap price. So that's one of the real strengths. And the other strength here that you'll note is the very small amount of DNA required. Some of the techniques for long read require an enormous amount of DNA, which in some cases is just not possible to get. So if you've got a small budget and a small amount of DNA, this is a very attractive option. Now, what we're able to do is assemble those reads in this case, and assemble those to give us a 96-fold coverage of the genome. And if we look at those, this is the technique that was used to assemble them. We've got a total assembly of about 880 megabases. Now, this is an 800 megabase genome, give or take, so we don't ever seem to have particularly accurate measurements of genome sizes. They change as we learn more about them. So as a first pass, this is a fairly complete genome. And you can see that that we had 5,000 scaffolds larger than 10 kilobases, and that covered almost all of the genome, 750 megabases. So this is by the standards, but certainly of the standards at the time, a particularly good, useful assembly. We have here, you can see that an N50 of 3.5 megabases for the scaffolds and 35 kb for the contigs. So that's what I think we'd expect from this platform. And a couple of things to note about it. This is a much better accuracy than we got from an Illumina short read assembly. You could just assemble Illumina short reads, but what we see here is a much greater accuracy from the MGI sequences when they're analysed in this way, with fewer mismatches and indels. So really a worthwhile approach to getting that assembly. And the BUSCO analysis showed us quite a high assembly for this degree of continuity. What it suggests is that, again, that the sequencing is quite accurate to be able to get a BUSCO that high. You wouldn't get that with a very error-prone sequence. So they're the basic statistics of the assembly. We have tried to push it further by seeing whether the long reads from Oxford Nanopore or PacBio can be used to scaffold or gap fill the genome, and that is quite successful. By adding here 1 million ONT reads, we increased the N50 from 35 kb to a megabase. This is a megabase and a contig N50, and the BUSCO came up to 93. And by using 674,000 PacBio reads, we were able to push the N50 to 1.6 megabases and the BUSCO to 94. So here we've got the advantages of the accuracy of the reads in this, but put together with a small amount of scaffolding from these other long read platforms. So there are a range of options there, and clearly there's many other options you could try. With many of the genomes, we've found it's combinations of technologies that have given us the result. And here it shows that the Sdlfr contigs can be assembled up more highly. We're looking at experiments at the moment to go further and try more options. We have been publishing a range of improvements in the sequencing and assembly techniques, so just look for papers citing that original one. The ongoing improvements that we're looking at are around DNA preparation. Many of the platforms, there's a specification for the quantity of DNA, the purity of the DNA, and of course, particularly for long reads, the length of those DNA fragments. You can't have the DNA degraded. Now, it does seem that this challenge is very different for different plant species because of the different contaminants that might be present. And some plants are particularly difficult because of various things that are in those plants. And quite often, it's very difficult to get all three of these in your DNA preparation. You can have plenty of DNA of high purity, but it's not very long. Or you can have long DNA that's pure, but you can't get very much of it. 2 out of 3 is usually relatively easy to get. All 3 is hard. So you've got to think about the platforms you use in those terms. So what we're working on a lot is developing new DNA preparation methods that will get the most out of these. And we're now looking at, we can't prepare better DNA to improve what we get out of the Sdlfr platform, because if we can get longer DNA of high purity, we think we'll get some advantages. Clearly, we see improvements in chemistry and, of course, in the software. The software can still play an important role in improving these platforms, and we've got more papers on that coming. So just to come back to the macadamia system, what we're doing, we've been talking about getting reference genomes, and we need those, but we're also interested in diversity, and that comes from resequencing. And here we're resequencing on the MGI platforms more than 300 genotypes of macadamia, and that's allowing us to get a complete chloroplast genome, and it's allowing us to get all the variants in the nuclear genome compared to the reference. And this will give us a complete platform knowledge of all known varieties of macadamia and a great survey of what exists in the wild. Effectively, a complete genomics platform for macadamia breeders. This is a growing and expanding industry, not only here, but internationally. But we'll really use genomics to accelerate the genetic improvement of this relatively recently domesticated species. We're extending this now to do the same sort of thing with citrus. 6 Australian wild citrus that have all sorts of traits, disease resistance that will be really important globally in fighting off the new major diseases around the world in citrus. We have resistance to those in the local species. We need to get the genomes quickly. And quite a lot of the wild relatives of our crops are present here in Australia. Australia is very rich, not in so much in crop species, but in their wild relatives. Really, with 10% of the plant biodiversity in the world here, we've got a lot of very important material. We're focusing on the wild relatives of rice, sorghum, macadamia, citrus, but all these other domesticated species, capturing that diversity by sequencing the genomes. We need to have methods for very cost-effectively sequencing large numbers of samples, and that's where some of these platforms are particularly useful. We're going back further. I've just come yesterday from 6 days in Western Queensland, where we're looking and working with the archaeologists on all the plants that exist in the desert and trying to explain what plants were used for food thousands of years ago, tens of thousands of years ago. Many of those species represent new opportunities for us to domesticate new crops or to obtain genes that we can use to help to adapt agriculture to climate change, and hence really play an important role in global food security. We have, of course, in Australia now an ongoing effort called Genomics of Australian Plant Species, which is aiming to ultimately sequence all of the plants. So this is 20,000 species. We need a lot of genomics capability. We need to think about how we come up with cost-effective ways of getting a reference genome for 20,000 species. And we need to look at how we sequence all the individuals. We've also got a threatened species initiative here. The nightcap oak, there's 90 known individuals with this species. It's a typical rare plant. We need to sequence all 90 of those. So we've captured all of the allelic variation known to exist for the species. So we need both the reference genomes and the resequencing. Another great example of where we're looking to apply this technology would be in sorghum. We've just been fortunate enough to be funded by the Carlsberg Foundation from Denmark to produce 500,000 mutants of sorghum. So here's the first 10,000 of those in the field at Gatton. We're moving ahead with 500,000 mutants. We'll have to sequence all of them, and this will be a major program, but this will give us a new platform for studying the genetics of this species, which we see as one of the key plants to support future food security, because it's adapted to dry and hot environments and could be the future cereal that will really be important globally. Huge requirement for efficient sequencing systems. Another example of how we're looking to apply this would be in more environmental genomics. Here we've looking at the sequence of a species in a transect from Melbourne up over the mountains and back to sea level, and looking at how the genome varies in different environments. What's the genome different? How is the genome different in the hotter and drier environments than in the cooler ones? That's giving us clues to the genes we need to be able to work with to produce crops for the future. And what we find is that it's not the temperature per se that matters with climate change, it's the change in the biotic environment. There's more pests and diseases when it gets hotter, and the plants need resistance to those. And that's the sort of insight we get by sequencing plants in the environment. And many cases, we want to go beyond that. We're looking to sequence everything in the rainforest to understand the dynamics of the rainforest. And this project here, which PGI has been helping us to address, is looking at the evolution of the rainforests and how species have come from Asia south and from Australia north, as Asia and Australia have come together in the last 5 million years, and looking at the dynamics and future of how we conserve rainforests and how we use the material in them. There is an enormous need for more of all of these platforms. I'll just end by saying we've got help to organise the TROPAG conference next year, but also the Harlan 4 conference, where we're going to look at the genomics of crop domestication. And that will bring the world to Brisbane to focus on these issues. And so there's a real opportunity for us to have a much greater focus on this sort of genomics happening here in Brisbane, Australia. I'd just acknowledge the research partners as well in this. BGI, of course, the Genome Innovation Hub here that's been really supportive of us exploring all of these options. And now, particularly the ARC Centre of Excellence for Plant Success in Nature and Agriculture, where we're going to be looking at using genomics to understand nature and to apply what we learn in agriculture. So I think I'll leave it there for questions. Thanks.
Thank you so much, Robert, for your talk. And we have 2 questions for you. So the first one is, if you were to start again on genome assembly with all you have learned through this process, what techniques would you implement for the best quality genome?
I think we'd still be implementing a range of techniques, and the techniques that we would implement continue to change and evolve. We're not in a static situation, and it depends very much on a whole range of things. It depends on how easy it is to get that DNA sample from the species of interest. Some species you really can't get the large amounts of DNA you need for some platforms, or you can't get the purity you need, or you can't get the length you need. So you've really got to adjust for that right at the start. Then I think no technique is best in all of those attributes. There are some strengths and weaknesses. I think the single tube long fragment method is particularly good for having a low cost and a low quantity of DNA required, and a reasonably high accuracy compared to other options. It doesn't necessarily give you the length, but it gives you all those other things. So, but I don't think any of these platforms are static. They're all improving all the time. So it's a continual appraisal. And we certainly, at one stage, we were evaluating every week which platform to use next, as things kept changing, because as soon as you evaluate one thing, something else changes. So I think it's a very exciting time to be in genomics because of all this, and we've got some great technology that's incredibly powerful to ask all sorts of questions, biological questions that we couldn't ask before. So I think that there's not an answer. It needs a whole lot more questions to be answered before you can specify that. And then, even then, I think we're still investigating new iterations of for example, this platform.
Well said. I agree with you, Robert, here. So the other question is from Yuri. The question is, deeper sequencing of a library or wider sequencing of various library, which is the better to get better genome assembly? So I'm splitting into 2 parts. Maybe I'll continue with the other one. So what is the ideal number of N50 reads per barcode for de novo genome assembly? So this person is using Sdlfr de novo as a assembler software. So Should I repeat the question?
Yeah, I'm not sure I'm the best to tell you how many reads per barcode or whatever. I think that probably you'll get that information directly from MGI, because they will know what the optimum is for that. And also to some extent the question about deeper reads or
wider sequencing. Various library.
Yeah, and I think that will be case specific. It will depend on the size of the genome and the complexity of the DNA sample that you're sequencing. Again, you have to make that reference, because there'll be advantages of both in different situations.
Yeah, if you have more questions regarding this, Yuri, maybe you can email us and we can try to get you best answer. So I'm not good at bioinformatics, so I also not sure what is the best N50 reads. Barcode is good for assembly. So, and one question I have for you, Robert. So which part of the plant you use when you are looking for high molecular weight genome?
Yeah, so
DNA?
It's most usual to use the leaves of plants, because they're usually relatively rich in DNA. It's very difficult, of course, to get DNA or RNA, which we quite sometimes have to get from particular tissues, because that's where the genes express from seeds or other parts of the plant, because quite often they're full of proteins or fats or carbohydrates, which are a problem. So it's really choosing a part of the plant that doesn't have those other interfering compounds, because a lot of things co-purify with DNA, like starch. So you want to avoid starch-rich parts of the plant. And you want to select a part that's rich in DNA, so large numbers of cells. But with leaves, even there, we tend to go for younger leaves, but relatively fully expanded, because some of the very young leaves have compounds that are protecting the leaf when they're young that actually interfere with the sequencing. So it's very much, you need to understand your plant, but not the very youngest leaves, not the oldest leaves, but the intermediate ones that have got good concentrated DNA. But you're really trying to avoid those contaminating secondary metabolites, particularly, that might be present at a particular stage.
Yeah. So when you are doing this comparative sequencing to all these different varieties, do you maintain the same age of the plant, or when you collect leaves, like the similar stages?
With most of the comparisons we've done, we've used exactly the same DNA sample. So that's same DNA sample.
Nice to know that.
So we use the same plant. So the great advantage of trees is you can keep them growing and keep taking leaves forever. So you can keep sequencing the same plant for hundreds of years. Probably.
Yeah, that's a good thing about plants. Especially perennial, like long.
And you can even go and sequence the parents if you know where they are, because you can have them growing at the same time. That's the advantage of working with forest trees. You can have several generations alive at the same time.
Nice to know that. And other thing is, just I'm curious, which DNA extraction kit you use for your? Is there any specific one you prefer?
I think almost every plant we've ended up with slightly different variations. We use sort of fairly standard approaches, but then we use standard alternatives and standard alternatives to those. For most, of course, short read sequencing, you can usually get very standard procedures. Almost any of them will work. It's only when you're trying to provide DNA for some of these long read methods that need a large amount of very long DNA. Sometimes we're trying to get the read. If we're trying to get a read length of 100 kb, you can't get that if the DNA is fragmented. You can only read to the end of the fragment. So there we are resorting back in some cases to using nuclear preps. So we isolate the nuclei first. But that's really a lot of trouble, and you wouldn't want to do that unless you actually had to, because it's hard to think of doing that on a large scale, doing thousands of samples. So there's the challenge there, and every species you've got to work through all the options. Even sometimes it's just a subtle difference in the temperature or the or the way you precipitate the DNA that really separates it from those contaminants that are very much specific to the individual species.
Mm-hmm. Mm-hmm. Nice to know that.
Yeah.
So I don't think there are any further questions today. So thank you all for attending this webinar, and thank you, Paul and Roberts, for your great talks. And I would appreciate if you could all participate in quick survey, which will start when you exit this webinar. So thank you so much, and have a good rest of the day.
Thank you.
Bye.
Thank you.
Bye.
See ya.
See you.